This is our last session of our meeting called Therapeutic Development and Gene Therapy. And our first speaker in this session will be Romas Geleziunas from Gilead. I practiced.
I was going to say.
And his title will be Exploring Combination Therapies to Achieve ART-Free HIV Remission. Thank you.
Wow, how sounds Peter. On a scale of 1 to 10, that's a 9. Very, very impressive. I'd like to first thank Hans, Peter, and Keith for inviting Gilead to present. We're very thankful for that. Okay, so let me take you through some of the things that we have been up to, and particularly a TLR7 story that's quite intriguing. We have a portfolio of agents that we have been exploring. It's actually not dissimilar from what Zach just kind of showed a few minutes ago, and I think maybe we should rename your lab maybe JZ Biopharma LLC or something, because we're talking the same thing here. You're just a little smaller, but we're doing the same thing. No, exactly. You're more nimble.
They'll trade you budgets.
Yeah. I'm not sure he needs it. I think you'll see. I think his stuff might be better.
So. Just remember, I'm sticking up for it.
Okay, so we've been going at this for about 7 years, this cure eradication program at Gilead, and we've spent a lot of time looking at histone deacetylase inhibitors, PKC agonists like Jerry. We've done high throughput screening, but the TLR7 story really emerged recently as being particularly interesting because we think it might fall into two spaces. It seems to be able to activate some virus expression, as you'll see. And it also might play a role in killing, and you'll see why that is. And what we particularly like about this is that it might be combinable with antibodies in the sense that this series of agents actually augments ADCC. So let me take you through what was essentially a 7-year journey. As you'll see, these things, they don't happen overnight. It takes a long time to build towards them. So actually, let's go back 10 years. So when I joined Gilead about 10 years ago, we launched this TLR7 program. And at the time, we were doing this because we were looking for a replacement for pegylated interferon for the treatment of hepatitis C. Back then, we didn't know that it would be dispensable. So we were looking for a way to minimize systemic interferon production and localize it in the liver. So that's the genesis of the program. But along the way, we've done a tremendous amount of characterization of these TLR7 agonists, and let me take you through that. So 9620, GS9620 is an agent that has emerged from multiple generations of optimization of these agents, and proves to be a very potent and selective TLR7 agonist. It's administered orally. It basically hits the TLR7 receptor, which resides in plasmacytoid dendritic cells as well as B cells. And in DCs, basically when you trigger this receptor with these small molecules, you elicit type 1 interferons, interferon alpha, to be specific in this particular case that we'll be looking at. So again, through the characterization of this compound, we discovered that when you dose this agent, 9620, or analogs of it, it basically has profound biological activity. It reduces surface antigen and viral DNA in woodchucks infected with woodchuck hepatitis virus, and also demonstrated antiviral activity in chimpanzees infected with HBV. So as a result of that, we decided to initiate clinical testing of this 9620 agent in HBV-infected persons. But the key is that we're trying to stay away from systemic interferon. So we basically dose to a level where we induce interferon-stimulated genes, ISGs, but not interferon alpha. So along the way, again, a lot of work by a lot of people led to some of these findings, which basically enticed us to want to look at this particular class of agent for HIV eradication as well. So in terms of kick, we kind of like this agent 9620 because we have seen it being able to induce viral RNA, viral particles from PBMC cultures from HIV-infected subjects on CART. And we've also been able to demonstrate that these agonists activate CD4 T cells. I don't know if these two are related. We're working hard to figure out if the activation of the CD4 T cell is in fact responsible for release of particles. We don't know that yet. But you'll see in the slides that I'll show you shortly that the CD4 T cell, it's only a very small fraction that gets activated in vivo, and the phenomenon is indirect. So basically the CD4 T cell only becomes activated in the presence or in PBMC cultures when dendritic cells are present. So there's some indirect way that you eventually activate CD4 cells. So for these reasons, we like it in terms of a viral inducing agent. On the kill side, well, we know from many years of work that TLR7 agonists enhance antigen presentation by dendritic cells, and they also activate CD8 T cells and NK cells. And as I mentioned earlier, we particularly like the NK cell activation, as well as some data that we've generated and others have generated with other TLR agonists that shows that these agents activate or enhance ADCC. So you can imagine the combination of TLR7s with antibodies. Now, the data at the bottom is basically an illustration of what we mean by activation of CD8 cells. Again, these are in PBMC cultures. On the left is the CD8s. On the right, NK cells. You get CD69 activation in PBMC cultures. And at the bottom, I note that we've actually seen this effect also in phase 1 studies. So this is not an in vitro artifact of some sort. It actually has been demonstrated in subjects that have been dosed clinically with this agent, 9620. All right, so this basically summarizes many, many years of work, and we've spent a lot of time on this trying to develop assays to demonstrate that TLR7 agonists activate HIV and SIV expression from various latency models, as well as PBMCs from HIV-infected subjects. And this is the best case scenario. This is as good as it gets. All right, so what you're looking at here is basically, as you can see, donors that are responsive. We have donors that are not responsive. We have donors that basically respond at the lower concentration of 100 nanomolar. We have donors that respond at the higher concentration. This is an absolute nightmare for someone in industry. Absolute nightmare. This is not dose linear. It's an inconsistent biological effect. This is not good. This is why it took us so long to actually move this even into in vivo testing, because, and we can't get a handle on this, we don't fully comprehend why this biology is so divergent from donor to donor. But it is what it is, and again, this is the best case scenario. It could vary from 50 to 60% responsive donors, and that's our reality in vitro, okay? So, nonetheless, after years of trying to convince ourselves, we finally did. We said, let's go do an in vivo experiment. Let's go test this in monkeys. And to do that, again, a tremendous amount of work was needed to set the foundation for that. First, we had to very carefully determine how to dose this agent. This agent is not benign, so you have to carefully do your dose ranging studies. You have to figure out the dosing frequency. And through a large amount of work, we figured out what the MED, which is the minimal effective doses to induce type 1 interferons in the periphery and ISGs in the periphery. We also determined that the best frequency of dosing was weekly or every other week. The second piece that we needed to develop is, many of you probably do know this, in the NHP community. The NHP community did not have a good suppressive antiretroviral regimen that could be given once daily, that was efficacious, that was well tolerated. We did not have that. So we enlisted the help of our colleagues in Gilead's formulation group. They are a tremendous group, very, very bright scientists, and they came up with essentially a formulation that enables the combination of tenofovir and more recently TDF, which is Gilead, combination of Gilead, FTC, and dolutegravir in a one injection once a day. So that's their contribution. It was very, very important. Okay, so here's the pilot study that we decided to do to test whether TLR7 agonists do anything in Rhesus macaques that are SIV infected. So 10 Indian Rhesus macaques, they were infected with SIV MAC251 intrarectally. They were given subcutaneous CART, the regimen that I just mentioned, and we started the CART at day 65 post-infection, so at the beginning of chronic set point. At day 320 post-infection, 8.5 months of CART, and highly suppressive CART. So again, this is, at that time, was without precedent, that you can actually do this for this long in a good, really good regimen. We initiated the dose escalation of not 9620, but a very close analog of 9620 in the dose range of 0.1 to 0.3 milligrams per kg. This agent is dosed orally in monkeys. 4 animals got the agent, and they received it every other week, and we gave them a total of 7 doses. Now, we dosed, in this particular study, 2 levels that induce peripheral interferon alpha. The placebos, there were 6. The primary endpoint for the study was plasma viral RNA rebound kinetics after stopping CART, but we also carefully looked at effects on plasma viremia during ART and immune activation. Okay, So let's take a look at the viral suppression. So here are the animals basically that initiated, as you can see, were infected, initiated ART, and after a period of time between 80 and 160 days, all animals became fully suppressed, and we initiated the TLR7 agonist dosing at day 320. All right, so let's take a look at some pharmacodynamic markers. I mentioned to you earlier that these agents indirectly activate CD4 T cells. Well, here's some in vivo data. As you can see, a small percentage of cells of CD4 T cells become activated upon dosing with the TLR7 agonist. It is transient. It returns to baseline after a while. And as you can see, this is a dose-ranging study. The first dose was 0.1 milligrams per kg, then 0.2, 0.3, and then the final 4 doses at 0.3 milligrams per kg. All right. Take a look at now CD8 cells. CD8 cells, slightly higher numbers become activated upon dosing with the 7 agonist in the range of 5 to 15%. Again, you see a trend towards dose linearity in the first 3 doses, and then the 4 subsequent doses hopping around the same level. We saw a similar pattern for NK cells, and we saw this, as I said earlier, in humans that were dosed. So we see very similar parallels here. All right, so now the intriguing data. So this is how the plasma RNA looked. So after the first 3 doses, we didn't see anything. Thankfully, we didn't stop the study after 3 doses. We kept on going. Dose number 4, we saw 2 of the 4 animals start to show copious amounts of RNA blipping. And as we continued dosing, 4 of the 4 animals blipped, and then again 4 of 4, and again 4 of 4. And if you look carefully, these are substantial amounts of viral RNA. They're ranging in the 500 to 1,000 copies per ml. So it was very intriguing to us to see this transient viremia, completely resuppressed, copious amounts. And so we were very excited about this data, as it was the first, I think, true demonstration that you can pharmacologically activate an AIDS virus in monkeys that are well ART suppressed. There's a note here at the bottom about sequencing, and I'm no expert in sequencing, but the experts told me that basically when they looked at the viral DNA in these cells, it was loaded with hypermutated sequences. But when they carefully analyzed the blips, there was no hypermutated sequences in the blips, which to them is consistent with the release of viral particles rather than cells lysing and disgorging just garbage DNA into the plasma. So this is a positive sign. Okay, so I told you that we designed this drug first and foremost to induce type 1 interferon for the treatment of HCV. Well, this is the primary pharmacodynamic marker, and often is asked of us, well, does your type 1 interferon induction correlate with viremia? And the answer, as you can see, is no, it doesn't. The first three doses basically induced type 1 interferon, again, to a nice trend towards dose linearity, and then the subsequent blips corresponded to again, plasma interferon alpha. So as you can see, there's not a perfect correlation here. It doesn't depend on the induction of interferon as we saw blips in the first 3 doses. All right, in terms of viral DNA, we took a careful look at that as well. We looked at viral DNA in PBMCs, in lymph nodes, and in colon, and that's what you're looking at the top. These are the TLR7 dosed animals compared to the 3 bottom panels where you're looking at the placebo animals. So you can see, again, there are intriguing trends. There was a reduction in proviral DNA in these PBMCs, but more so in the lymph nodes and in colon in 3 of the 4 animals, clearly not all animals. But there seems to be a difference between these animals and the placebo-dosed animals. All right, so what happens when we release the ART? So I told you we suppress the animals nicely. That's the middle part over here. I told you about the three doses that required to initiate 4 blips over here in the subsequent doses. And then we stop the ART over here and we let the rebound take place. As you can see, we did not cure any animals. But there was a little bit of an interesting observation here again. The animals in blue, the ones that received TLR7 agonist, had a slightly lower set point, about a half a log compared to the animals that did not receive the drug, which are depicted here in red. So again, an intriguing observation. We don't quite know what the mechanism is, but it seems to be something else that seems to be positive with regard to this compound. All right, so just to summarize this portion really quickly for you. So in terms of TLR7 agonists in vitro, or in ex vivo in vitro, we have seen over the years activation of HIV expression in PBMCs, though the effect is inconsistent. It could vary from as low as 50% to up to 70% of donors that we've looked at. The expression can be twofold or up to higher than that. In this particular group of patients that I just showed you, 72% of them responded in terms of a superior to two-fold induction, and the mean was about nine-fold. I showed you that with these TLR7 agonists, we're able to activate CD8 T cells and NK cells. This is indirect, and there is donor-dependent variation, as I mentioned. In terms of hypotheses or correlates, we don't have any. Our best hypothesis is, as I mentioned, this drug induces type 1 interferons, an antiviral factor. So we kind of think that there's a balancing act here. In donors that particularly induce very good type 1 interferon responses may mask the viral blip or the viral induction. Others may tilt in the other way. So it's the best we can come up with. We really don't know why we have this variation. In terms of in vivo, I showed you that we have induced transient plasma viremia for the first time with a pharmacologic agent in monkeys that are truly well suppressed for a long time. We've induced transient activation of CD4 cells. I don't know if the activation of CD4s and HIV, or SIV in this case, are linked. We're working on that. We activated CD8 cells and NK cells, induced a variety of cytokines and chemokines. And there was a trend towards a decrease in DNA, viral DNA in lymph nodes and colon, and we seem to have reduced the viral load set point after stopping CAR. Well, this is a small study, and at Gilead we tend to be very, very aggressive. We didn't wait much longer. We decided to go to the clinic and find out if this animal model has any predictive value and any of this could work clinically. So we don't have the results yet, but I will show you the design of the clinical study. It's a small pilot study. Again, the design is it's a single to multiple ascending dose. So if the patient tolerates the single dose, they continue to the multiple dose portion of the trial. There are 3 dose cohorts. It is placebo-controlled, randomized, and double-blinded. The study population is 24 HIV-infected adults. They are virologically suppressed for over 12 months on CART. The study treatment is 3 doses of 9620 at 1, 2, and 4 milligrams, or placebo. Now it's 1 tablet orally once a day. So these doses are the same doses that we're testing in HPV-infected patients as well as we're testing in healthy volunteers. So we have a good deal of experience with these doses of this particular compound. The 9620 to placebo ratio is 6 to 2, so 6 individuals get drug, 2 get placebo. And the schedule is 1 dose every other week times 6 doses. Again, we're replicating the monkey study here. So this is good. I mean, we're actually using the monkey trial as guidance for the clinical study. We're very happy about that. The key differences, however, from the SIV Macaque study that I just showed you is that the dose escalation is by group, not by subject. And that could matter. We don't know at this point. And there will not be any discontinuation of ART. In terms of the primary objectives, it is first and foremost safety, as you can imagine, looking for adverse events and looking at clinical laboratory assessments, CBC, transaminases. We are going to look at virological activity, of course, by TACMAN. Secondary objectives include pharmacokinetics, pharmacodynamics, single copy assay just to make sure in case we are getting activity, viral activity, but we can't pick it up with COBOS. We're going to look at single copy. We're going to look with the sensitive cell-associated RNA method as well as other assays. All right, so that's the TLR7 part. So the thing that we've been asking ourselves for a while now that we've seen those data, is what would TLR7 agonists combine well with? And I mentioned, I think, on a few occasions already that we think that maybe HIV antibodies, broadly neutralizing antibodies, that bind envelope would be a very nice pairing here. And the reason is depicted on this slide. If a latency reversal agent like TLR7, or even through homeostatic proliferation or antigenic stimulation, we could induce good amounts of GP120 on the surface of infected cells. This might be a handle for an antibody to latch onto and then recruit a variety of potential effector functions mediated by NK cells, macrophages, and complement. So this is the concept, and we already have some evidence in vitro that this might be working, so we're eager to test this in vivo. And the reason we are thinking about this is because of the recent work of a number of groups that have identified and cloned these very nice new broadly neutralizing antibodies that are more potent and more broad. The VRC, Michelle Nussenzweig and Dennis Burton, to name the key players in this field. The antibody that we're particularly interested in is PGT121, circled in blue. It binds the V3 glycans. It's very, very potent. And the reason we're interested in this particular antibody is because of the work of our close colleague Dan Baruch, and Dan showed this work this morning. I want to just recap this because this is really intriguing. When we saw this data, we became quite excited. So Dan basically did a study in the upper left-hand graph where he combined antiretrovirals, again, the ART regimen that I told you about, with PGT121. This is the human antibody. And he compared this to the right graph, which was ART alone. And he told you this morning that you basically get a nice suppression of viremia. And what he also mentioned that was really key to us is that the animals that basically showed a temporary period of viral remission, he told you that these animals all eventually rebounded, but the animals that showed the temporary period of viral remission by plasma RNA are also the ones that basically showed the best reductions in viral DNA in PBMC and GALT. And this is very dissimilar from the situation that you see here with antiretrovirals only. You do not get any temporary remission period, and you also don't get any declines in tissue-associated viral DNA. So this, to us, suggests that perhaps these antibodies are doing something more than small molecule antivirals. Maybe they are getting into tissues, maybe they are binding to cells and basically lysing these cells. So this is why we're so intrigued by this. So as a result of this study, Gilead obtained an exclusive license from Biosciences, who own the intellectual property estate for these antibodies, and our goal is to help improve these antibodies and to develop them. So we launched an internal lead optimization program. The purpose of the program was to define which functions of the antibodies work best to clear viral reservoirs, kill cells, and to engineer these bodies with superior enhanced effector function. So to give you an update on that, we have just recently selected the clinical candidate, and so in 18 months from now, we will have it in clinical testing, and hopefully the TLR7 and the antibody will eventually merge in the clinic in 2 years or so. I also want to note that we're collaborating with Dan Baruch. Again, it's a very nice collaboration with the Gates Foundation and Dan Baruch. So Dan and Gates are basically optimized, or have not optimized, but are developing GMP material for PGT121, the parent molecule. So what we're going to have ultimately is the testing of the parent molecule 121 and a Gilead optimized antibody. So we'll see which one will fare better, and that hopefully will guide the field, help guide the field, and both antibodies will be tested clinically. Okay, so to wrap up for you, so what I told you today is that I think we might have our hands on a new latency reversing agent. Maybe that's the wrong name for it. Maybe it's a viral-inducing agent. It might be activating T follicular helper reservoirs. We don't know at this point. We'll have to figure that out. But anyway, it seems to be in vivo inducing good amounts of virus. We are thinking of combining this TLR7 agonist with, as you heard, anti-HIV envelope antibodies. We're also exploring bispecific antibodies, and we also have some collaborations on therapeutic vaccines and combinations with the TLR7. The hope, obviously, and then this TLR7 might be able to modulate some of these functions, including ADCC. And obviously, the hope is to reduce these reservoirs, and obviously, this would be done in the production of CART, so we don't reseed reservoirs. So let me just finish by acknowledging some key people. So Bailey and Jill Hattersley, these are the two brilliant young ladies who came up with the CART formulation. Again, tremendous work. Their second generation of CART is really tremendous, less viscous, very efficacious, so they did a wonderful job. Angela, Libalu, Jasmine, Jeff, and Derek are the ones that are working on the in vitro aspects of TLR7. Tomas is my close partner for the last 7 years working on this cure program. John Corbin is the fellow who headed up the lead optimization program that led to the clinical candidate antibody that we're bringing forward to the clinic. Tiffany and Joe work very closely with me and work very closely with James Whitney, who did the monkey study, the TLR7 study. Of course, special thanks to Dan Baruch, close colleague. We work a lot together, very, very productive collaboration. I want to also acknowledge Jay Lalazari and the various donors who have provided cells for these assays. Both this concept and other concepts would not be possible without being able to test these agents in patient cells. Dennis Burton, who is the one who identified PGT121, and Christine and Po Ying at Theraclone, again, great collaborators, though they handed off the antibodies to us. So I guess I'll stop here and take questions. Do we have time?
Thank you very much. Yeah, there's time for a few questions. Mike.
I'm sure you've heard this before, but it did look like on the graph where you were showing the viral loads and the suppression, that your red controls took longer times in general to suppress than your blue blue positives. So one could easily tell a story about that even in the unseen part of that, you're having a greater replication application of that and receptor differences.
Yeah.
What do you say?
Thank you. That's a great question. So what we did is, you're absolutely right, very good, keen observation. So what we did is we compared per animal, post and pre set points. And by doing it per animal, it still comes out to be half a log. But thank you for that. That's a very good observation, which is why we went back to individual animals. You don't want to look at it just this way, in other words.
Florian? Oh, sorry.
I think it would be very interesting to actually continue the TLR7 delivery, because you have 3 time points where you give it. At each time point, you have the same kind of viral blip and the same magnitude. So the question really comes up, are you essentially reseeding the reservoir by activation? And so I think it would be interesting to go longer and see whether those blips are actually starting to decrease or disappear or not.
Yes. Thank you for that question. So we have an ongoing study where we have gone to 20 doses. So I can't discuss that just yet because it's ongoing, but thank you for that. That's exactly right. Carefully looking at where this is going. In terms of reseeding of the reservoir, I don't think so, because when we looked at viral DNA in tissue, it's in fact dropping. So I don't think we're not expanding the reservoir, that's for sure. But I don't know if we're actually putting a dent into it. But stay tuned. We'll release those data. In that second study, in fact, what we did is not only did we go to 20 doses, we also dropped the doses to ISG levels, because this is germane to the clinic. These doses are too high for dosing in the clinic because they induce type 1 interferon. But at ISG levels, what will happen? This is the intriguing question.
Jerry?
Did you look at levels of antiviral T cells or NHPs or anything? This would be consistent with little vaccination beds.
Yes.
Which would be consistent with dropping your viral loads.
Not in this study, but in the follow-up study, we are looking at that. That's a very good point. Thank you.
One more?
This is a similar question. How do you interpret the drop in set point? Do you think something else is going on, or is the impact of the reservoir change?
I think Jerry from JZ Biopharma, see, he thinks like a. I think maybe this is it. Maybe we're eliciting an adaptive immune response that, during these many rounds of vaccination, if you will, maybe inducing a CD8 response a bit more robust. We activate B cells. I haven't talked about that, but we have to carefully look at what happened. And TLR7 resides in B cells. We may be changing the humoral response, which may also be helping contribute to control this. So a lot needs to happen here to characterize this, but it's intriguing.
Let's do one more question. Davey, back there.
Are you planning to look at
Autoantibodies. Yeah. Yeah, no, it's good. Yes. I hear you. No, it is good. Not in this small pilot study, but it has been raised actually. Your point is a good one. And as we go forward, the first study is obviously just to see if we can cause blipping, right? It's a very simple study just to confirm that this has any merit. And if it does, we'll do more studies, and then we'll say again. Well, again, it's all the materials have been accounted for. It's a tight, small study, so I presume if we have plasma, or we can go look at that, but it's not the first priority at this point. Again, small study.
Okay, I think we need to move on. Thank you very much. Next presentation will be by Jen Adair, also from the Hutch here. She will be talking about development of a globally portable platform for lentivirus-mediated hematopoietic stem cell gene therapy to treat HIV.
I'll hold it, yeah. It's this keyboard or that one? Okay. All right. Well, thank you guys for coming back after lunch. And I hope in the next 15 minutes I'll kind of keep everyone awake. I'm actually really excited to talk about the work. I don't think I'll have many opportunities in my life to say that the data you're about to see was generated in 9 months. So I'd like to take that opportunity. I also really enjoyed the CAB poster last night that Martha presented. And one of the things she had was this little booklet that defines terms that are not necessarily commonly understood by the community. And so I wanted to simplify this very scientific title to reference something Matt Porteous said yesterday about manufacturing cell and gene therapies in a box. And the data I'm about to show you is a tangible step towards that end. I am not an HIV biologist. I'm a stem cell biologist. I came to the Hutch in 2008 to work with Hans Peter on translating clinical trials of stem cell gene therapy for other diseases, namely glioblastoma multiforme and Fanconi anemia. This is a list of all the diseases that are successfully treated with stem cell and T cell gene therapy currently in the clinic. And coming soon, as many of you have heard, are advances in treatment for HIV and sickle cell. But when we start talking about these diseases, there's a pretty obvious elephant in the room. This is the global distribution of HIV infection in adults as of 2014, and here are the centers that are capable of manufacturing gene-modified cells. We have a very big distribution problem, and part of that is because of the complexity of this process. You just heard Jess talk about, and you heard investigators yesterday talk about how we have to treat patients and then take blood products out of the body, isolate those cells, then we have to select specific cell types, then we have to manipulate those cells by hand, which is exposing them to the air and the environment around them, and then we have to test those products to make sure that they're suitable for infusion back into patients. And this is a pretty intense process, and it has to be conducted under FDA standards called Current Good Manufacturing Practices, or GMP. For stem cells, we can do this in about 2.5 days with current technology. T cells, it's somewhere between 5 and 14 days. And so these are pretty long periods of time outside the body. Certainly, if we could get these gene transfer elements into a syringe that we could transport around the world, that would ideally be cheap and portable. There are two major issues that we have to resolve, though, before that's going to be possible. And the first of those is that we need to make sure that we can get enough of our therapeutic genetic element to the cells that we're interested in. When we're injecting something into the body, we're kind of putting it in the presence of all of those cells, and we need to make sure that enough of the cells we want will get that gene product. The second issue is that if the gene product does go into cells that we don't want it to go into, is that safe? And we can overcome those barriers, but it's going to be many years, I think, before we're there. And so what Matt alluded to yesterday was if we could condense this process into this kind of black box, where we're still doing cells outside the body and transferring the gene in a very small format that's amenable to clinics around the world, we might have a little faster approach to portabilizing gene therapy. These are the 6 features that a system would have to be able to do this reasonably. And until a couple of years ago, the technology to do this was not available. For different reasons, I was introduced to this device produced by Miltenyi Biotech called the Clinimax Prodigy last summer. And it's the second generation of that stem cell separation device I showed you in step 3. It was designed to combine steps 2 and 3. So instead of that person manipulating the product up front to get ready to select the cells you want, now all of that would happen in this device in kind of an automated fashion. And that meant that this device was capable of separating cells, it's capable of spinning them down, it's capable of incubating them at specific temperatures, all the things that we want it to be able to do for gene transfer. And so we hypothesized that we could actually advance this methodology to be able to produce these gene-modified blood stem cells, in our case, in a box, in a laboratory. But the onus is on us to demonstrate 4 basic principles to prove this. 1, that we can isolate stem cells and we can isolate enough of them, that we can efficiently transfer genes into these cells, that these cells are safe for infusion, and also that they're functional once they go back into an immunocompromised host. And I hope what I'm going to show you in the next few slides is going to prove to you that we're capable of doing that. So we started with human-mobilized apheresis products because this is what the device was designed to purify initially, and that's what we have the most experience with. This is the product that would most commonly be used in patients who are adults. So we had healthy donors mobilized, and we collected apheresis products. We loaded these products onto the device, and we initially separated them. This is an immunomagnetic column that's built into the device, and we used basic CD34 separation. We then performed culture and transduction in the chamber on this device. The anti-HIV vector that we used is the same vector being used in Hans Peter's clinical trial for treatment of patients with AIDS lymphoma. It has the same anti-HIV cassette you just heard Jeff Bartlett discuss with 2 anti-HIV transgenes, but it also contains a chemotherapy selectable gene called MGMT, which Chris Peterson referenced yesterday. For the gene therapists in the room, most of these transduction conditions are familiar. I will say that we do not use retronectin in these chambers because we wouldn't be able to get the cells back out. And then we take the product that comes off of this device after the automated process, and we test it to make sure that it would meet the same criteria that our cell products going into gene therapy trials would have to meet. And then to prove it's functional, we put it into mice that have been struck by lightning. Glad you're all still awake. I don't know why this is our universal symbol for our UV or for total body irradiation, but it is. These mice get sublethal irradiation to show functionality. And to build this onto the existing programs took development of 5 additional custom programs for this device, and the tubing set, which is this kind of winding plastic tubing and bag set that you see up here. We actually had to use 2 different ones provided by the company because they didn't at the time make one that connected to all the different parts of the machine that we needed it to. And what we were able to show is that we could very efficiently isolate CD34 positive cells within the same clinical parameters that the first generation system is capable of doing at really high numbers, which is highlighted in blue, and then the percent yields are in red. We were also able to efficiently transduce these cells to the same level that we would see in a hand or a manipulated process. I'm showing you the results from 2 different donors here. On the left, you're looking at just cultured cells over an 11-day period at 2 different time points. I will point out that this second donor actually has a higher level of gene transfer than we would typically like to see in a gene therapy trial, but I'd rather have the problem of figuring out how to dial that back than have the problem of generating it when it doesn't exist. And then this colony forming assay, and I'll show you more data like this throughout the talk, is basically an assay where we simulate conditions in the bone marrow and we ask, are these cells capable of forming little different blood cell types? And we can pick those colonies and look at how many of them accepted the gene transfer, and we see good percentages of gene transfer in this process. We used the exact same testing criteria under GMP conditions to evaluate these products that we manufactured, and they met all the same criteria that our hand-manipulated products should have to meet, indicating they were safe for infusion. And they engraft into, again, mice struck by lightning. So the first donor in black, the second donor in blue, this is just looking at the percentage of cells in the blood that are human. And this graph is showing you how many of those human cells in the blood and the bone marrow at 12 weeks were gene modified. And it's not surprising that the blue donor is a little higher because that was the one that had the higher level of gene transfer. Now, apheresis is used, like I said, for adults. If you're a pediatric patient or you're a patient whose health condition doesn't allow you to be treated with growth factors, we would get those stem cells from bone marrow. So we also wanted to see if we could use this process to do the same thing from a bone marrow starting product. But there are a few things that we have to do a little bit differently. In addition to the fact that you undergo a bone marrow harvest, which we did from 2 healthy donors, this particular process, we need to remove red blood cells first because they interfere with our ability to select stem cells. And we do this through a process called head of starch sedimentation, which is shown up on the right. Again, we isolate CD34 cells. We use the same transduction conditions and vector that I just showed you. This required us to generate 2 more custom programs for the front-end processing that didn't exist on the device before, and we had to use the same 2 different tubing sets to accomplish this. I'm not going to go into all of the data. I'm just going to show you the 34 enrichment at this point, and then I'll briefly discuss the other endpoints. We didn't get as effective CD34 enrichment. It's kind of within the range, above, around 50% that would be considered normal, but it's pretty consistently low. So we can definitely try to improve those programs, I think, to get better stem cell yields from the bone marrow. If you were paying attention to the blue line for the apheresis products, we're looking at about a 10-fold reduction in the total number of stem cells here. We were still able to transduce these stem cells on the device and transplant them into mice. I can tell you that they transduce efficiently. We saw the same variation in the amount of virus that went in. We did have 1 donor who had a higher level that we'll have to dial back. We were able to confirm that these products met infusion criteria, and we were also able to show that they engrafted in immunodeficient mice. But if we're going to talk about pulling this strategy in to really assess whether or not we have an effective anti-HIV therapy, we need a model where we can measure resistance to HIV or something like it. And I'm here in Seattle. I work with Hans-Peter Kiem. I'm going to start working with monkeys. We decided to do this in the pigtail macaque. I'll show you in this particular experiment, we give the animal growth factor, G-CSF and SCF, for 5 days, and then we harvest bone marrow. It's a little bit different than what you saw for the 2 human products, but this is what we standardly do. We then put this on the device. Again, we had to remove the red blood cells first, and we used our head of starch sedimentation program to do that. In terms of the selection, the human reagent to select stem cells doesn't cross-react with the monkey. So we had to develop a modified version of that protocol, which required us to do it in 2 steps. 2 more programs, now we're up to 9. We were able to successfully do that, though. And then, I'm going backwards instead of forwards. This time when we did the transduction, we added 2 doses of vector instead of 1, so it's a higher overall amount of virus. And we didn't use the anti-HIV vector. We wanted to make it very simple to see that these cells went in and did what we wanted them to do. So we used the same vector backbone, just expressing a green fluorescent protein. So now we can look in the blood for green cells and know that we got engraftment. Again, we took those products, and this time we infused them back into the animal after a myeloablative dose of irradiation. So this would be lethal if these blood cells are not capable of reconstituting blood cell production in this monkey. We did this in 2 different animals. Like the human bone marrow, we saw subpar CD34 enrichment, probably because we started with bone marrow and then added a second step to the selection. But because monkeys are small, these produce really efficient cell doses still, so we were able to go forward with the transplants. We saw very efficient gene transfer by the number of blood cells that expressed GFP. These cells were also capable of forming colonies, and those colonies did have the gene successfully transferred. One measure of how well these cells can perform is how quickly they recover the blood once we transplant them back into the monkey, and I'll draw your attention to these 2 columns on the far right. So the neutrophil recovery, 8 and 12 days, platelet recovery, 23 and 14 days, is pretty much what we would hope to see with the cell doses that these monkeys received. So by these measures, they're behaving the same way they would as if we'd done this entire process by hand. The real question, do they have gene-modified cells? And the answer is they do. So the solid line that is green granulocytes. The dashed line in open squares is green lymphocytes. We can see these cells emerge in these monkeys over time. They persist. We did see 1 drop in the granulocytes in this monkey that corresponded with a taper of some immunosuppression these animals get, because GFP is not a monkey gene. We increased that dosing, and we could see that gene-modified cell level go right back up. We don't know why it was only restricted to the granulocytes. It didn't affect the lymphocyte compartment. But to use this model in the anti-HIV approach, we want to make sure that we can get gene-modified lymphocytes, so we also looked at that. You're only looking at GFP-positive lymphocytes here, and you can see that early after transplant, the majority of the lymphocytes are B cells. But within about 100 to 150 days after transplant, we can definitely see gene-modified T cells of the 3, 4, and 8 lineages. Now, the punchline of all of this is that it didn't take us 2.5 days. We actually did it overnight. So all of the processes I just showed you were 30 hours or less, which is a significant improvement in our ability to do this, I think, clinically. So in summary, we've been able to demonstrate the proof of principle that in a box, ex vivo, lentivirus-mediated gene therapy is possible in stem cells. We've done this with minimal requirement for additional equipment. We can show that we can use multiple initial products. We got sufficient CD34 enrichment, but we definitely think it could be better, especially for the marrow products. We got a very efficient gene transfer, total process less than 30 hours. The amount of time the cells are exposed to the virus that transfers those genes is only 12 to 14 hours. And all of these products showed suitability to be infused into an immunocompromised host. So what are we going to do next? We want to see if we can expand this to T cells. We need to optimize this process a little bit more, and I'll talk a little bit about that on the next slide. Of course, the true proof of principle would be taking this on the road and showing that other people can do it too. And we definitely want to validate this in an HIV model like the non-human primate. So for the community, what does this mean? It means that we now have something tangible that can lead to global distribution of anti-HIV cell and gene therapy. Should you be excited about this? Yes, but I would just, not jump quite yet. As I mentioned, we had to do a lot of modification to this device. And these are just two fun things that I like to show. If you walked into a clinic and saw that bag on the left full of binder clips, I don't think you'd be very impressed with the technological advance in that particular clinic. But this was some of the stuff that we had to do initially to figure out how to make this work. Some of the stuff we were able to fix, so we were able to get a funnelized bag manufactured from a company for these sedimentations. But they only make it in 2 sizes, and it's not perfect for the different kinds of cell products you get. We're working on that. This glorified bag holder that you might recognize does not come standard with the device unless you live in a tropical climate between the months of November to February, and then she can be provided. That we still have to address with the company in terms of getting some of the things that we need to make this move forward. And with that, there are a couple of people who happen to be in the room that I need to thank too. I want to make special mention of Hans Peter, who convinced me in the first year of my faculty position here that this was worth my startup money, and I think he was absolutely right. I also like to thank my technician, Autumn Lee, who assisted me in all of these experiments and sometimes served as the bag holder. Dan Humphreys, who helped with some of the clonal assays that I didn't talk to you about today. Kevin Hayworth and Devika Chandrasekaran in Hans Peter's lab, a postdoc and talented technician. They're both talented, that helped with these projects. And of course, the non-human primate techs, Veronica, Erica, and Kelvin. I also need to point out Tim Waters from Miltenyi Biotech. When I sat him down and said, I want to do this with the device, he was like, this is going to take a lot of programming. And I said, okay, so what's the problem? And he said, you're right. So he just worked on it with me and was very patient in working with me to get us through this process. I also need to thank Shelley Heinfeld in the room, who provided us with those apheresis products I talked about and the guidance during the development of this project, as well as all the cell production facility staff here who put up with me dancing around in that lab with this device. And then Grant Trowbridge at WSU and Guy Sauvageau, who provided some small molecule for these particular studies. And with that, I will take any questions. Yes.
So, as David Baltimore said at the community forum the other night, nothing in science should be impossible. I also think the technology around these issues shouldn't be considered impossible either. Several years ago, one of the things you kept hearing in cure research was that it's got to be made globally accessible. Well, everybody understands that it has to be globally accessible, but that's not a reason to stop progress in the science. And I think what we're seeing with this wonderful advance to me is that kind of technology and the technology for it at the same time we have science.
Thank you.
Thank you.
I saw another, Guillermo.
Really cool stuff. I was curious about the increase in viral marking that you saw. It kind of looked like it was reproducible at the time. Is there something about the machine or the way the machine is doing the transduction that you think might lead to increased viral marking, or do you have any ideas?
Yeah, so I kind of glossed over it, but in one of the things that we put in the media is a small molecule called UM729. And this is a pyrimidindole derivative that was developed by Guy Sauvageau in Montreal. We have subsequently removed that in some manual experiments, and we can with the same donor cells, and we can show that it drops the vector copy number. In the 2 donors where we had high copy number, so to give you guys a reference point, 5 is the highest we would want to go in terms of units in that result. One donor had 10, the other donor had, I think, 11. When we take that molecule out, we can get them both to 5. So we're kind of right at the limit. We can reduce the viral MOI that we add. The trick is, the other donor was kind of in the normal range. Do we want to reduce MOI for everybody and then maybe have lower than optimal efficiencies in some? I mean, certainly there's a lot of room to play around with it, but I think we have the platform to do that now.
Do you see more variability between donors using this machine than you would see the kind of standard of care GM type manipulation?
It's difficult to compare because they are really 2 different processes. I think we're just starting to gather the information on donor-to-donor variability in this process. And we're pushing this machine to do stuff it hasn't done before. We're doing it in ways that it's not typically done manually. We're jerry-rigging it, as you saw, in some cases. So, when we did the apheresis products and we saw that second donor result, I thought, well, maybe we just got really good, really fast. But then when we did the bone marrows, and the first one turned out that way, and then the second one was lower, it suggested that we probably really don't understand all of the things that are going on there. So we still have work to do, but I think we've made a big step in the right direction.
Okay, thank you very much.
Well, we've come to the end. So thank you to all of our speakers and the poster presenters, our community members, and everyone who participated and stayed to the very end. It's super. This meeting represents a lot of work, and very little of it was done by me. So I want to thank Tom Andrus, Mary Lamoury, and Jacob. These guys have real day jobs, and they do them very well, and it's amazing that they can pull off a conference like this. It's wonderful. I want to acknowledge our scientific organizing committee. Again, Paula Cannon, Julie Overbaugh, Ron Mitsuyasu, Eric Seelbach, Jeff Simmons, Jerry Zach, and Galit Alter. Thank you to our platinum sponsors, Amphar, Gilead, and Sangamo. Our silver sponsors, Calimmune, Life Technologies, Miltenyi, Biotech, and Sigma Aldrich. We would love to hear your thoughts and feedback on this. Do you find this conference useful? If there are particular things you liked about it, please email me. If there's things you didn't like, email Hans Peter. And with that and the right feedback, we would love to see you here at our next conference. Thank you very much.